Okay, so there is a one important topic, that is suspension of pharmaceutical suspension is a full dispersion. So in which the internal phase, whatever it may be therapeutically active ingredient is dispersed uniformly. So there are two phases, external and internal, and that was dispersed phase. It is uniformly throughout the external phase. So they are consisting insoluble solid particles having a range of at least, uh, I mean, uh, that was starting from 0.5 to 5 in micron size. And the external phase, that was also known as suspending medium, is generally aqueous in some instances, but maybe an organic or oily liquid for a non-oral use. So basically, they are classified like a based on the size of solid particles, colloidal, porous, and similarly nanosuspension. Uh, there are differ in sizes. Like colloidal suspension is less than 1 micron. Porous suspension is more than 1 micron, and nanosuspensions are less than 50 nanomicrons. Uh, so advantages are like they are used in drug suspension, exhibits the higher rate of bioavailability, and uh, also used in a solid dosage forms. So basically, there is a disperse mean solid particles. So it is directly related to both solid and liquid formulation. Uh, what are the formulation consideration are like a solution, suspension, capsule, compressed tablets, and coated tablets. And suspension can improve chemical stability both in circumstances and in a certain drug. So controlled drug release suspension can mask the unpleasant bitter taste of drug, and there is a easy in administration. While we always having a patient compliance, many patients complaining about their taste, like a bitter taste, metallic taste, salty taste, sour taste. So it can be directly inhibit or can be mask the unpleasant form of the taste of the drug, and that's why it is in administration, is in patient compliance, and in every situation this can be a ideal dosage form. And if there are advantages, similarly also in a side, both sides of point, disadvantages are also physical stability, sedimentation rate, ज्याला आपण बोलतो sedimentation rate and compaction can cause various problems. There are various disasters. It is bulky in nature, so having needs to be a sufficient care must be taken while during handling and transport. Also difficult to formulate because there are very challenging to maintaining the sizes in the microns, nanomicrons, less than 50 nanomicrons, more than 1 micron. So, and also the solid particles are, first of all, you have to sterilize, you have to maintain all hygienic conditions, you have to maintain all good manufacturing practices, good laboratory practices, good compounding and manufacturing practices. So it is very difficult to formulate. Like there are weighing procedures, there are various sieving procedures, size reduction, size separation processes. Uh, so meanwhile, it is difficult to formulate. Also, it is uniform and accurate dose cannot be achieved unless suspension or packing in a dosage form. Currently, बऱ्याच वेळेला आपण जेव्हा टॅबलेट जर आपण घेतोय, इट्स अ सॉलिड फॉर्म, इट्स युनिट डोसेज फॉर्म, त्याच्यामध्ये क्लिअरली डोस किती आहे तो मेन्शन असतो. बट इन दी सस्पेन्शन अँड इमल्शन्स अँड सिरप्स, dose is obviously there is a prescribed, but if there are upper and lower meniscus type, so that is cannot be achieved. Whatever the dose we required, whatever the accurate dose we required, that cannot be achieved, and unless suspension are packed in unit dosage form. So these are the all disadvantages. Also, there are features desired in a pharmaceutical suspensions, like the suspended particles should not settle rapidly and sediment produce. So as we all know, in every situation we have a kind of features, so must be easily resuspended by the use of moderate amount of shaking. That's why in every solid liquid formulation there are uh, guideline given, there is a direction given, shake well before use. But coincidentally that direction is cannot be recommended or cannot be amendment. It should be easy to pour it, not watery and no grittiness. So there are no grittiness because there are solid particle also that will be minimize the grittiness of the particular liquid medium or particularly solid medium also. It should have pleasing in odor, and also having a color and palatability. Uh, Also having good syringeability. So whatever the suspension we are going to formulate or whatever suspensions we are going to manufacture, that are specially prepared for parental formulation. Like there are two classes. One of the most and major important classes are there. First one is LVP and another one is SVP. LVP means large volume parentals and SVP means small volume parentals. Both having a differ. Small volume parentals are from 1 ml to 100 ml. And that large volume parentals are starting from 100 ml to till 1000 ml, both having hypotonicity and isotonicity. Hypotonic solution, uh, there are a special mint of preservatives are added in uh, large volume parental and not in a small volume parental. So these are the proper, kind of difference between SVP and LVP. So you're getting my point or not? So as we all know, if you are going to check about the evaluation parameter of the volumes, so small volume parental having 100 ml or less than 100 ml, and large volume parental having 100 and 1 ml starting to ranges between 1000 ml. And the route of administration for small volume parentals are IV, IM, and SCSLD, intravenous, intramuscular, and subcutaneous. For large volume parental, they are intravenous and yes, it is only applicable or restricted or curriculum for intravenous. Dosage needs are like small volume parentals are both available in vials, ampoules, like a single or multiple. But in a LVP, that is PSA solutions, like a saline bottles, that is only a single form. Preservatives are used only in a large volume, small volume parental, not in a large volume parental. Buffers are used in small volume parentals, not in a large volume parentals. So both preservative and buffers are used only for small volume parental. Getting my point or not? Then again, there is a isotonicity. So it was essential for large volume parental, okay, not for small volume parental. Even it is must in to add in that large volume parental. And yes, of course, pyrogenicity is required for large volume parental because it is in a large form. And if your material, your formulation, your parental formulation, your suspension is contaminated or it is containing pyrogens, so it is very dangerous to the health of the patient, whatever you are going to administer to intravenously. And yes, use of small volume parenteral are therapeutic and diagnostic. But while large volume parentals are generally, we are always see that these are used for a nutritional purpose, detoxification purpose, and during surgeries. So that is the difference between small volume parental and large volume parental. Now we are going to see that another features of pharmaceutical suspension that should be easy to pour, not watery, and not bitterness, should be have pleasing odor, color, and palatability, good syringeability. It should be physically, chemically, and microbiologically stable. Parenteral ophthalmic suspension should be sterilizable. So, that kind of features always anyone can see in your formulation, your whatever formulated. Uh, if you are going to see another part of suspension, like a formulation consideration for suspension, so there are a suspending agent, various suspending agents are there, like carboxymethyl cellulose, CMC, gums, like gum, tragacan, guar gum, Indian tragacan gum powder, acacia powder, sodium alginate is there, wetting agents, polysorbate 80, acacia, tragacan is there. Uh, if you are going to see about buffers, so pH adjusting agents is there, phosphates, carbonated buffers. If we are going to see about osmotic agents or dextrose, mannitol, sorbitol, coloring agent like amaranth, titanium oxide, preservatives like diazonium acetate, butyl paraben, propylene glycol. As we all know that paraben family are generally majorly used preservatives. So in the form of suspension, we have to use butyl paraben only and propylene glycol. And there are two types of manufacturing for suspension, like dispersion method and precipitation method. Uh, these are like here. Suspension can be made by using two methods, namely dispersion methods, precipitation method. This method is divided again into three kinds: precipitation with organic solvents, precipitation with a pH change of media, and precipitation with a double decomposition. So there are another part also. We can and we have already discussed it about in our next last video. So evaluation of suspension, there is sedimentation method, rheological method, electrokinetic method, and micromagnetic method. Okay, so whatever the another part we will see in next lecture. Thank you.